Good day, Kavibal, and welcome to our YouTube webinar series, Research Presentation Special. For the discussion today, the topic will be on potential of bioluminescence Vibrio SP from Gaza Al-Klamis as biosensor of copper and zinc. Before we begin, take note of the following reminders. Make sure you are registered to the webinar to have your e-certificate of participation. Visit certificate.vibalgroup.com to generate your proof of attendance. Place your questions in the comment box allotted during the session and they will be addressed by our speaker later on. Share the video using hashtag Learners1PH as our official hashtag to all our Vibal webinars. Experience learning, Kavibal! And now, to proceed with our webinar this afternoon, it is my pleasure to introduce to you our distinguished speaker today. Ms. Lorraine Joyce M. Del Rosario is a former teacher at the University of Santo Tomas Senior High School and instructor at the University of Santo Tomas. She has taught fields of science and practical research and has also been appointed as the moderator of La Stampa, the official publication of the UST Senior High School, and as an e-learning specialist and educational technology coordinator. She was a member of the UST SHS Executive Committee and some academic-related societies and organizations in the biological field. Apart from graduating as valedictorian in the special program in journalism in high school, Ms. Del Rosario earned a BS Biology degree from the University of the Philippines, Baguio in 2017, supported by a DOST scholarship. Her undergraduate thesis with her partner and advisor was awarded as one of the two best paper presentations in the third Philippine Solid and Hazardous Waste Management Conference. She is pursuing her Master of Science degree in Biology at UP Diliman granted with DOST-AST HRDP scholarship. Her current field of interest is in genetics, particularly in the study of DNA in molecular phylogenetics. Aside from being an educator and a graduate student, Ms. Del Rosario spends her time writing, tutoring English online, accepting speaking and hosting engagements, and traveling as a hobby prior to the pandemic. Ladies and gentlemen, let us all welcome Ms. Lorraine Joyce M. Del Rosario.
Hello, good afternoon to each and everyone. And thank you very much, Ms. Jillian, for the very warm introduction. So this webinar is special because we will be having a research presentation. And you know how research has impacted our lives greatly. You know, the world has come a long way, right? Ever since the introduction of research into the society. How did it influence you? How did it influence us? You know, the products of today are simply the products of research. The comfort that we enjoy, every little thing that contributes to the sustenance of life is brought about by extensive research. And I can't think of any other great contribution of research other than our fight against coronavirus, which is still ongoing. You know, though the light at the end of the tunnel still seems unclear, research has helped us progress through studies of vaccines and the nature of the virus. You know, whatever is the settings now, we can still make research possible anytime and anywhere. And in this virtual mode of delivery, we are still capable of producing research. We're still capable of producing data using online sources. Or actually, we can make comprehensive or systematic reviews, meta-analysis, or even in silico studies. So in silico studies are performed via simulation on the computer. And with research, we continue producing great people, such as our scientists, researchers, and the like, who are actually considered unsung heroes of the world. The purpose of research is to enhance society by advancing knowledge through scientific theories, concepts, and ideas. And our research purpose is met through forming hypotheses, collecting data, analyzing. Now, the goal of research should always be apparent. So join me today in creating and presenting our research. So thank you very much to the Vibal Group for always giving us this opportunity. So this is a very special segment in Vibal right now, and here we will talk about research. Actually, my presentation is divided into two parts. Prior to the actual presentation of my research, we will be discussing some guidelines, important information, some inspiration or motivation on how to come up with research ideas, how to conduct a research, or how to actually write and present a research. And then afterwards, in the second half of our webinar presentation, I will discuss an example of a research, which is actually my undergraduate thesis about bioluminescent bacteria. So it will be a very interesting field. It will be a very interesting topic. So I hope you stay tuned with me. Without further ado, let's have the first part. So how do we really come up with research ideas? I am a science major and I do not want to actually sound biased with quantitative researches. So I made it more general. So this is applicable for both quantitative, qualitative, or even mixed methods of research designs. So how to come up with the research ideas? Let's say someone gave you a present, right? But you're not allowed to open it. Perhaps you're wondering, what could be inside the box? I want to know what's inside the box. So in an attempt to guess the item, you try to make ways. You try to lift it up, move it around, or probably simply ask the giver, what's inside? The drive of wanting to know something is a representation of what we call curiosity. So in this case, your main question is, what's present inside the box? And for you to satisfy your curiosity, you attempt to get information by doing things that can probably give you clues, like what I've mentioned. Shake the box, try to guess what's inside, try to ask. So the probing for truth can represent what we call inquiry. So in its Latin etymology, inquiry, literally translate to into asking or seeking. So inquiry and research are often used interchangeably. Both terms aim to search of knowledge and information, search for knowledge and information about a problem. The difference between research and inquiry is a process and how to search for the truth. So research uses investigation that gives actual evidences or facts collected and analyzed in a systemized process, more specifically, what we call scientific method. So looking at our example, we inquired about what is the content of the present or the gift. For the reasons it did not follow the scientific method and fundamental characteristics of research, we're not satisfied. So learning and research is a way of obtaining knowledge about your surroundings. It's a learning process for you to obtain knowledge or information about people, things, places, or events. And actually, it can be defined in many different ways. And oftentimes, it's defined based on characteristics. So there are two main authors who defined research. One is Shuttleworth. He mentioned that research includes any gathering of data, information, and facts for the advancement of knowledge. And another information or definition is from Creswell, wherein research is defined as a process of steps used to collect and analyze information to increase an understanding of a topic or issue. And actually, you can decide whether to create a basic research or an applied research. What's the difference between the two? So basic research desires or aims to expand knowledge. So there is a drive for curiosity and it intends to answer why, what, or how questions. So it increases understanding of fundamental principles or theories. And it doesn't have immediate commercial objectives. So it may not necessarily result in an intervention or a solution to a practical problem. Sometimes you just, or most of the time, you just want to gather information, gather knowledge. Whereas for applied research, new knowledge acquired has a specific commercial objectives. So you can create products, procedures, or services. And it answers specific questions aimed at solving practical problems. So you can decide whether to pursue basic research or applied research. But whatever it is, a good research starts with a good research question or thesis statement. So it takes into consideration how to formulate a good research. And there are three general characteristics. So, of course, it should detail the problem statement and further describe and refine the issue under study. It should help support a focused, arguable thesis and construction of a logical argument, and should dictate the valid and acceptable methods to be able to conduct the research. And if you satisfy these three general characteristics, for sure, you have a room for tangible or conclusive results. And you should exactly know the position of your thesis statement. It should reflect the type of paper you are writing. It should be concise because otherwise it will be very disorganized. And you can place it at the end of your first paragraph. So how do you develop this research question? So you can identify subjects of interest, or you can reflect on studies that were done in the past. You can improve on them. You feel like there are still some gaps or some lacking information. And then you can brainstorm a problem that will lead to few implied questions. If you already have that, you can do some preliminary research. You can Google. And of course, with Google, you try to search for credible references and determine information gaps. Now, with those, you can narrow the scope and focus of your research, and it could already lead to the next steps, creating hypothesis, evaluating the question, and understanding the implication of your research, especially in the long run. So now that you have a good research question, how do you conduct a research? If it's a quantitative, most especially, you can make use of the scientific method. So asking a question, doing some background research, constructing a hypothesis or an educated guess. And then of course you would like to test that hypothesis, whether is it working or not. Yeah, if it's not working, probably you can troubleshoot. If it is, then it's a new finding. We can analyze data, draw conclusions. You have to make sure that the results will answer or will align with the hypothesis. And of course, if it doesn't, it's still a finding. You can make use of references, valid references, to further make your claims stronger. And from there on, then you can form new hypotheses and experiments again. And the best thing, or one of the best or important thing you should do after the research is to communicate results. So that's why we publish papers, we present research presentations, we write manuscripts. So in order to communicate the results to the readers or to the audience around the world. So there are a lot of topics which you can choose from, not limited to science. You can have different fields, different area. You can have real world phenomena. Yeah, and then just formulate a good research question and proceed on to the next steps until such time that you arrive at a conclusive finding. Yeah, and there are also factors which can be controlled or not controlled in research, such as for planned work, inputs, activities, and outputs, and intended results such as outcomes or impacts. So of course there will be a lot of challenges, right? And for now, maybe one of the challenges is how to do or how to experiment in the field or in the laboratory now that pandemic is still ongoing, and usually we have some lockdowns, we have some quarantine protocols, but it shouldn't stop you there, right? We can actually still do research even at home. So a while ago we have opened up possibilities of doing research, right, even right in the comfort of our very home, and it's possible to conduct research online. So we can choose our topic, determine fields of study, list items that interest us. We can narrow it down further, search online databases, research what experts has to say. Of course, fact check the information. It's very important. Organize this information, create the bibliography, and update your outline and fill in the gaps. So anything is possible. Like right now, we're having our webinar presentation remotely. So anything is possible so long as we really have the determination to pursue it. So when we conduct online qualitative research, we can make use of these methods of gathering data, right? Web interviews, phone interviews, mobile diaries, online bulletin boards, etc. So if there's a will, there's definitely a way. And just a brief on how to write and present a research. It's important to understand the structure of a research paper. So this research paper outline components are, these are the usual or the norm, beginning from the cover page abstract, table of contents, intro, methods, results, discussion, conclusion, bibliography where you put your references because we want to avoid plagiarism, and appendix. Or another illustration to show it. The sequence of IMRAD, introduction, method, result, and discussion, is very common parts or are very common parts of a research paper. Each section addresses a different objective, and the research question should always be the heart of the paper so that we can have a guide, from the research question, what do we want to find out? What do we want to answer? That's why we're doing methodology procedures. So the introduction, what is known already, we can introduce key players, and then we can identify what is unknown. So that's the gap that we want to fill in our research. We can make some rationale and hypothesis. So how and why should we fill the gap? And what are we going to do? So that is in our methodology. And afterwards, we have the results. What are our findings, discussion? How do the results bridge the gap? We can use resources to explain our findings, especially credible resources. And conclusion, what does this mean for us going forward? So some brief overview of each section. So of course, we have the background, which includes the purpose and reasons. We can make use of the inverted pyramid style, which is from general to specific. So this serves as an overview on how we can write our background. So we can contextualize, put the problem into context, conceptualize, point out the gap in the knowledge, and resolve, identify the problem to be addressed. There. And objectives of the study, we can remember the acronym SMART. Specific should be specific, measurable, attainable, realistic, and time-bounded. Of course, it should be time-bounded. We don't want our research running along for many, many years, right? We need to find results. We need to find data. The significance will identify who will benefit from the research and how will they be benefited. And of course, what is the contribution of our study? And the scope and limitation determines the coverage. Identifying all the things that also will and will not be covered in order to be clear and specific. Another important foundation of research is RRL. So it requires summarizing, paraphrasing, and writing indirect speeches. Your skills here are needed. So this is the a very exhaustive analysis of materials related to our study. So it also prepares us. It categorizes our sources. It can help us screen relevant and irrelevant information by evaluating literature sources. And we can also search for gaps as we review or related literatures. This is a very popular post, like a meme on Facebook, right? You cannot cram the RRL. So I see many friends sharing or posting, commenting on this, that they're saying, "Watch me, I can do it. I can cram the RRL." Yeah, actually, okay, you can cram the RRL, but the content or the accuracy, the quality of your work is compromised, is affected. So basically, RRL takes time. So you will review, you will really read a lot of relevant and credible literature, which can help you in your topic. Other structure will include methodology, of course, results, and the discussion. So the methodology should include the design, the population, sample size, techniques, data gathering procedure, data analysis procedures, and some statistical treatments. If you're doing experiments, you need to include field site, description of instruments, yeah, which I can demonstrate to you later when I present the research. Detail also in the method how you will manipulate the data or that you assembled to get at the information that you want in order to answer your question. And of course, include some statistical tools. So here are some of the tips. Be explicit, be clear, be clear about the focus, be as detailed as possible about the proposed work, be specific about the means of evaluating the data, be certain with the connection, and of course, be realistic with what can be accomplished. Chapter 4 is the results and discussion. So results are made of the facts, figures, information that were discovered during the research. And discussion are the drawn ideas from these facts, figures, or information. So it should be coupled with literature review. So this is the reason why you're also doing exhaustive analysis, because you will need your information from the literature review into the discussion. So outcomes from doing the methodology is the result in discussion. And yeah, of course, it will screen the data, analyze the screen data, and you can make use of visual aids. You have to really connect it with your lit review. And so the conclusion, finally, will restate the main idea and research objectives. If you were able to answer the research objectives, what are the main outcomes, and you can suggest further proceedings and new prospects. So that's why recommendation is also part of this chapter. So it's used to call for actions or solutions to the problems that you have investigated. And as I've mentioned, researches should be time bound. Usually you can create some Gantt charts or timetables in order to help you visualize the timeline and the progress of your research. And in presenting the research, of course, it's important to communicate, right? Communicate your results, your findings. So that's why we present research papers. And our main goal is not to impress people, but actually to express what we have found. So we can make use of great. We should focus on great content and fewer slide, high quality graphics and images, good contrast of colors, uniform theme, and consistent fonts. So we are designing minimalized slides in order to highlight key points and data of presentation. So we are communicating our results. So we need to make sure that we are highlighting the hotspots. We know what we're going to say, and it can be understood by our audiences, especially if these are general audiences. Our pictures, figures will really help a lot because they paint 1,000 words. And of course, we can always do rehearsing when before presenting. So yeah, that's the first part of this topic. Now, on the next half is the presentation of an actual research paper. So this is our research topic in 2017, along with my thesis partner, Mira De Jesus, and our thesis advisor, Ma'am Ofelia Heron, PhD. So this is the potential of bioluminescent Vibrio SP from Gaza Aclamis as biosensor of copper and zinc. So this is under the field of microbiology and environmental science. If you're wondering what Vibrio SP is, it's a bioluminescent bacteria. And Gaza Aclamis is a fish. It's actually sapsap in common terms. So as an introduction, probably you have already seen pictures of this, or perhaps if you have experiences in the ocean, what do you think is the secret of the glowing sea? Why do we have this amazing light reaction or bioluminescence? Yeah, so actually these are attributed to the bioluminescence capability of organisms, specifically some microorganisms. Have you ever seen images like this? So jellyfishes which are glowing in the dark, or some fish which have some parts which are glowing. Yeah, so there's a possibility that uh, microorganisms like bioluminescent bacteria can attach to these organisms, and it makes them glow. It's a very beautiful sight, especially in the evening or especially in the dark room. Yeah, and you will really marvel and appreciate nature. So it's the phenomenon known as luminescence. When I say luminescence, it's the emission of light by an object which do not result from heating, but it's an emission of light when the temperature is below that of what we call incandescence. So when you extract, for instance, a part of that ocean or water, you put it in an Erlenmeyer flask like this, and you go to a dark room, a very, very dark room. It appears like this. So this is bioluminescence, and it's resulting from biochemical and enzymatic activity of living organisms, including certain bacteria. And those are what we call, the Vibrio species are popular species. Vibrio is a popular genus of bioluminescent bacteria. It's most abundant and widely distributed. It can also be found in terrestrial, other than the water. Yeah, it can be found in fresh water and marine. Can be found on the species that are in the fresh waters and marine, and they're free living in the ocean. That's why you can notice they are on the surface of the water or on the surface of organisms. And actually, they have ecological and biological rules. They could be as gut symbionts in the digestive tract of marine organisms, could be parasites in crustaceans and insects, light organ symbionts in teleost fish, and saprophytes growing on dead fish or meat. So when we isolated bioluminescent bacteria from samples, and then we swab, we made a strict plate method on this one. Yeah. And then observe it under dark condition. This is how it appears. Actually, we can create there's some bioluminescent arts which can be created from this. So it's actually very interesting. So how do bioluminescent bacteria exhibit this bioluminescent reaction? So this is also what we want to really emphasize in our research. So in bacteria, we have what we call LOX operon. And in this particular bacteria, the LOX operon involve the genes LOXCDA-DEG. So LOXA gene and LOXB gene are the luciferase gene, which codes for the alpha and beta subunits of luciferase. Luciferase is the enzyme responsible for the light reaction. Whereas the LOXCDE genes are fatty acid reductase genes that codes for the polypeptides. So the enzyme luciferase catalyzes the conversion of aldehydes to fatty acids and oxidation of a long-chain aliphatic aldehyde and reduced flavin mononucleotide FMNH2. That's why you can see it in the reaction here. So it requires oxygen, of course, and the excess energy is liberated in the process and emitted as a luminescent blue-green light at 490 nanometer. Yeah, so actually, it's a blue-green light if you're going to see it in actual. And bioluminescent, yeah, so this is also another illustration showing it. So this is the operon, LOXCDAB. So these are the genes in an expression vector. So the insertion of these into the organism allows the fatty acid reductase enzyme complex and luciferase to be expressed. And the function of luciferase confers the organism the ability to emit light. So for instance, these are two bacteria. This one is a non-luminescent bacteria like E. coli. So it doesn't have such kind of ability. But for bioluminescent bacteria like Vibrio, wherein they can harbor the bacterial luciferase and fatty acid reductase genes, they have the ability to emit blue-green light. So yeah, that's bioluminescence. So it's emission of light by an organism and or by a laboratory biochemical system derived from an organism, like the ghostly glow of bacteria. Yeah, so yeah, this is another way to show it. So this is the reaction, and this is the operon. And interestingly, bioluminescent bacteria grow uh, exhibit the bioluminescence better if they are many or if the quantity is higher. So this is what we call the quorum sensing. So bioluminescence in bacteria is enabled through autoinduction or what we call quorum sensing. It involves the self-production of a diffusible pheromone called an autoinducer. This serves as the extracellular signal that accumulates in the medium. And it's the one responsible for the characteristic response from cells, which is light emission. So thus, bioluminescent bacteria can only exhibit bioluminescence when a certain high cell density is detected through the system. So for example, if it's just a single-celled organism like this, it will not cause the bioluminescence. So this autoinducer binds to the receptor of the bacteria, and upon binding it, it enables the DNA to encode for those LOX genes a while ago. And the LOX genes code for the luciferase, which catalyze the reaction that leads to the emission of light as shown in this figure. So when the cell growth continues, the level of autoinducers in the media also start to accumulate in this confined environment. And a very low intensity or light emission can be detected. But what if there is a high level of inducers or autoinducers? So it could activate easily the luminescent systems of the luminous bacteria in the medium. So this regulatory response to the autoinducer leads to the induction of the expression of the genes in the operon. So this is what we call as the quorum sensing. And in the title, we've mentioned that bacteria from this fish can actually be biosensor of heavy metals, copper, and zinc. What do I mean by biosensor? When I say biosensor, these are analytical devices that are based on the union between biological and physiochemical components. That's why bio plus sensor, right? So it's a blending of exquisite selectivity of biology with the processing power of modern microelectronics and optoelectronics. So it means using biological organisms or components with the sensing activity. For example, some biological components could be the antibodies, enzymes, or tissue slices, which will be used to recognize and interact with the analyte. And physiochemical components or transducers are magnetic, colorimetric devices, optical, amperometric, and potentiometric. So these are used to convert the interactions into signals that are amplified with respect to the concentration of the analyte. So integrating it with the topic. So as this biosensor, it could actually be a self-contained integrated device that is capable of providing specific quantitative or semi-quantitative analytical information. So basically, the enzyme here or the biological component is the bioluminescent bacteria. And the bioluminescence that it can produce, it can be under the physiochemical components. So a sensor, this sensor integrates biological element with the physiochemical transducer to produce this electronic signal, which is proportional to a single analyte, which is then conveyed with a to a detector. So basically, the use of bioluminescent bacteria as biosensors is efficient because the bacteria are reproducible. We can culture them. They're sensitive to changes in condition, and they provide a cost-effective way for the detection of pollutants, which are the heavy metals. For example, if certain parameters are not within the optimal range for the bacterial growth, this affects quorum sensing, right? Subsequently affecting the bioluminescent capability of the bacteria. So compared to other bacteria, bioluminescent bacteria can be readily seen in the environment due to their luminescence, and other bacteria do not have noticeable characteristics. So it's really difficult to determine, for instance, if there is pollution or contaminant in a certain area. So it can also be illustrated in this figure, right? So this is the principle behind the movement of the analyte or the component. And here are the components there. So yeah, this is an abstract of our presentation or our research topic. And basically, our objective is to isolate the bioluminescent bacteria from the fish, the Gaza aclamis, which is the sapsap, and to analyze the potential of this bacteria as bioreceptor or biosensor of copper and zinc ions in water samples. So it's a new trend in biosensors for water monitoring. So our sampling site, so we are already in the methodology. So our sampling site is in the Lingayen Gulf. So this is the Lingayen Gulf in Luzon with its coordinates there. So this gulf is traversed by Agno River through the Limahong Channel, which is considered to be the mouth of the river. Now, the river has its headwater in the mountains of Cordillera, east of the Gulf. Now, because of this connection, heavy metals that originate from mining activities in the upstream Baguio and Cordillera mining district located at its headwaters are carried off in sediments and could add to the pollution downstream. So sediments which are sloughed off from the mines contain heavy metals in relatively high levels, particularly trace elements of copper, cadmium, lead, zinc, and mercury. So there are literature proving this. It's the literature from McManus and Thea Eng in 1990. So since heavy metals are among the most serious pollutants, there's a need to develop rapid and sensitive biomonitoring methods, right, for heavy metals in the marine environment. So at high amounts, of course, heavy metals can become toxic to marine organisms and they can pose risk to human health, especially if fish are contaminated. So how did we know that there is presence of heavy metal in the water sample? So we collected water sample from Lingayen Gulf, Limahong Channel. And we also tested physiochemical parameters such as pH, temperature, salinity, in order to add information to our discussion. So the water samples were brought to the laboratory, and with the help of flame atomic absorption spectroscopy, they were able to find out presence of heavy metals there. So we want to find out presence of the copper and zinc. And where did we isolate the bacteria? We isolated it from the fish, the Gaza aclamis. So inoculation, isolation of bioluminescent bacteria from specifically the fish eyes, gills, and guts. And then, of course, we need to purify this BLB through strict plate method. We also inoculated this BLB into broth medium of 5% saline solution in order to also monitor or examine the bioluminescence. And then for us to know how it will be effective as biosensor, we also prepared this is already the experimental setup. We prepared different concentrations of copper and zinc metal solutions. So we prepared this by serial dilution and we incorporated it into the broth medium. We want to find out if it will affect the quorum sensing, if it will affect the light emitting activity or the bioluminescence. So we prepared and we put different concentrations of copper and zinc metal solutions and incorporated it into the broth solution. So we assess the biosensing potential by measuring it using the Fluostar Omega. So it's a device which is used to measure bioluminescence. And after that, the data, of course, we need to statistically analyze the data. We analyze it through SPSS. And we also wanted to determine we also wanted to determine the identification of the bacteria. So we did a molecular identification through 16S rRNA gene sequencing. We analyzed, we aligned, assembled the sequences through softwares, and then we had to compare it with available sequences online through BLAST. And that's how we were able to search for the species of the bacteria that we have isolated from the fish. So this is the fish, the Gaza, there. Gaza aclamis, or the sapsap, right? Commonly known as the sapsap. So we swab the gut, the eyes, and the gills, eyes, gut of the bacteria, and then we put it on the strict plate. We use the strict plate method. There. And then from there, so this is the result of the bioluminescent bacterial swab. There. So it means there is the presence of bioluminescent bacteria. So you can imagine, right, from the cotton buds a while ago, we swab it on the Petri plates through stick plate method. And then we have to, there's a time for this actually. We have to wait for quorum sensing before we can finally observe this glowing effect from the medium, proving that we have isolated bioluminescent bacteria. And here are the results. For the results. So we wanted to find out, right, if there is a biosensing ability. So this figure shows an inverse relationship between bioluminescence and concentration of the copper and zinc measured over a period of 30 minutes. So this is relative light units, RLU. This is time, and the legend. Yeah, this is our legend. So 0.01 ppm, varying concentration of the heavy metal. So in this case, this is zinc. And this is copper. All right. So copper and zinc. Anyway, the trend is the same for both copper and zinc. So the two figures show an inverse relationship between bioluminescence and concentration of copper and zinc measured over this time period. So the decrease of bioluminescence with different concentration of heavy metals is actually associated with the metal chelating activity. So metal chelating activity damages cellular metabolism, which can further lead to cell death. So in the reaction of bioluminescence, wherein flavin mononucleotide and carboxylic acids are the products, dissociation of the luciferase FMN carboxylic acid complex follows in an ordered manner. So this activity involves carboxylic acid functional groups which bind to the available heavy metals in the surrounding. So it means the more metals there are, more carboxylic acid functional groups are bound, and thus they are unable to produce light. So the higher the concentration of the heavy metal, the lower is the relative light units. The lower is the ability to produce light. So it could be a biosensor. And then this is also, we also measured percent bioluminescence inhibition. So this is a comparison of percentage inhibition between copper and zinc. This is copper, this is zinc, measured also after 30 minutes of incubation. So statistical analysis through t-test as well, assuming equal variances, generated this generated a two-tailed p-value of 0.251, which is higher than the critical p-value of 0.05. It only means that the effects of copper and zinc were not significantly different from each other. Nevertheless, there's just a slight difference, and this could be attributed to the kind of the metallic species. So zinc has an inhibition higher for every concentration compared to copper, which may be because zinc is more reactive than copper based on activity series of metals. And a more reactive species of metal is able to displace other metals bound to a chelate when present in high concentration. So yeah, so this is how we communicate the results. We, upon gathering data, upon gathering our findings, we try to also, or we compare it to the references, or we search for possible explanation in the references in order to be able to discuss our results. And finally, This is the molecular phylogenetic analysis of Gaza acclamis and the BLB, the bioluminescent bacteria that was isolated. So it's inferred using the neighbor joining method. So this is through 16S rRNA sequencing. So this is the bacteria UPB BLB05. It goes to show that it is in the Vibrio species. The square sequence is best pairwise aligned with the 16S RNA sequence of Vibrio natriagens, which is a free living bacteria with the fastest generation time known. So that's the result. It only goes to show it could prove that Vibrio sp can be a potential receptor in a biosensor for copper and zinc, as proven by the inverse relationship of bioluminescence to heavy metal concentration, and can therefore provide basis for risk assessment of heavy metal contamination, especially in bodies of water. And for recommendations, to make further studies even more conclusive, even more effective, we can perhaps it's better to have a prolonged duration of the experiment to really determine the effect of the heavy metals. And we can make use of longer periods of time. I hope I hope the future researchers can identify taxonomic classification up to the species level, and to be able to have a fabrication of a biosensor in which the abilities of the bioluminescent bacteria can be utilized. Yeah, so sorry, it's already mentioned there. Yeah, so that is the presentation of the research on the potential of bioluminescent Vibrio sp from Gaza acclamis as biosensor of copper and zinc. And we also need to be careful, right, with answering our objectives, with answering our research questions. We hope that we will be able to design methodologies that will really lead us to the answers, or really lead us to the conclusive results. And if ever your findings are not as what you have or what you have expected, it's not the end of the world. It's still a result. Okay. And you can actually make use of references or literatures online to possibly explain those. So, just for some key takeaways, research is to see what everybody else has seen and to think what nobody else has thought. So, supposing is good, but finding out is better, and we can do that with research, right? So, thank you very much for listening, and hopefully, even in this time of pandemic, we can be able to produce researches. Thank you for listening. That is it for my presentation. If you have questions, you can also reach me in this email address. And here are the references for my slides and for my presentations.
Thank you very much, Ma'am Lorraine. Now it's time for our question and answer portion. So let's look at our YouTube comment section if there are any questions for our speaker. So all our viewers, if you have any questions regarding the research that our speaker have presented, or you have any questions with regards to creating and presenting a research, you may comment it in our chat box. Alright, I think our viewers doesn't have any questions because of the clear and concise discussion of our speaker. Do you have any reminders for our viewers na lang po, ma'am?
Yeah, thank you very much, Miss Jillian. And actually, if you don't have questions for now, that's all right. It's understandable. But if you have some questions after our webinar today, you can always reach me in my email address. And I'd be very glad to answer those queries. And so research has really paved ways on how to improve our lives in all aspects. So it's actually the foundation, also a good foundation of the society. So I really hope I was also able to inspire our audiences, our viewers, about the importance of research and how we can actually conduct researches on our own. We have to look for good resources, good references that can help us in our research, and also some good mentors. Yeah, some good mentors, some institutions which can also help us, because research can also be expensive and costly. Therefore, we can requests for aids or for assistance, especially financially speaking. So anything, so long as you know you have a dream, you have the determination to pursue a certain study, nothing is impossible. Yeah, that would be all. Thank you very much, as always, to the Vival Group for opportunities like this.
There we have it. In behalf of Vibal Group Incorporated, I would like to thank our speaker for today for this very eye-opening and insightful learning session. It is always an honor to have you, ma'am. And to all our Kavibal viewers, all thanks to you for your continuous patronage to all our daily learning session. Don't forget to
Thank you.
register to get your e-certificate of participation. The link is in the caption of the webinar. We also encourage you to subscribe and watch on our official Vival Facebook and YouTube channel. Muli, maraming